Hello everyone and welcome to our module on bacterial identification. This slide shows the algorithm to identify gram-positive bacteria, and you have to memorize this as a medical student, and there are a lot of ways to do it. The way I used to do it and the way I recommend is to just write this out over and over again until it sticks in your memory, either on a piece of paper or on a whiteboard. But by whatever method you use, you're going to have to memorize this, and you're going to have to know how to identify bacteria based on the results of common microbiology tests. So we're going to go through how you identify the bacteria in this module and talk about how each of the tests work. So once you identify that a bacteria is a gram-positive bacteria, you then want to look at the shape. There are three shapes you may see. The first is cocci, which are little circles. We'll see those in a minute. The second is rods, and then the third are these long branching filamentous shapes that look almost like fungi. Those are very unique, and those are only seen in Actinomyces and Nocardia. Once you identify a rod, there's only a couple of choices of what it can be, but if you identify a cocci, there are lots of different options for what the bacteria are going to be. So you need to do several microbiology tests to determine what species you're dealing with. The first test you do is called the catalase test, and it's either positive or negative. Catalase positive organisms are staph, and catalase negative are strep. Strep also tends to occur in chains, whereas staph tends to occur in clusters. You can further subdivide the staph by the coagulase test. So if the coagulase test is positive, that's Staph aureus. And you can just remember Staph aureus, which is a very common bacteria, is coagulase positive and catalase positive. It's positive for both of the C tests. Coagulase negative staph is one of two things. It's either Staph epidermidis or Staph saprophyticus, and you can separate those two out by using a Nova-Bayesian sensitivity test. Now, if you're dealing with a catalase negative cocci, you're dealing with strep, and the primary way we differentiate strep, once we know we've got strep present, is to look at how it grows on blood agar. So there are certain strep bacteria that are so-called beta hemolytic. Those are bacteria like strep pyogenes and agalactae, and you can separate those two from each other by the bacitracin test. There are other strep that are alpha hemolytic. Those are like strep pneumonia and strep viridans, and you can separate those from each other by the Optochin sensitivity test. And then finally, there are strep that are gamma hemolytic, which is actually a misnomer. These strep are group D strep, and they don't hemolyze at all. And those are bacteria like Enterococcus and strep bovis, and you can separate those from each other by seeing how they grow on bile or high sodium chloride concentration media. So now what we're going to do is go through this in more detail and talk about each of the tests used to distinguish the bacteria. Let me first mention something called Lancefield grouping. This is a system for classifying only streptococci, so not for staph or other bacteria. And it's based on C carbohydrates that are found in the cell wall of streptococci. And when these are present, they will agglutinate specific antisera for that C carbohydrate. And there are lots of commercially available tests for the different C carbohydrate antigens. And there are three clinically relevant groups to the practice of medicine. Group A strep is the strep pyogenes group, Group B is strep agalactae, and Group D includes Enterococcus. And I mention this now because you'll commonly hear that certain strep are Group A, Group B, or Group D, and you should know that they come from this Lancefield grouping. So cocci are a very common shape, especially for gram-positive bacteria. They're little circles like this, and they tend to come in certain patterns. Sometimes you'll see diplococci, which are pairs of cocci. Some cocci tend to appear in chains like this. That's what streptococci tend to do. And some tend to grow in bunches or clusters. That's what staphylococci tend to do. And on this slide, I've shown you some pictures of how these things look under the microscope. So these are streptococci here, and you can see that there are long chains forming. Over here we've got staphylococci, and you can see that there are little bunches together of these small circle bacteria. So blood agar is a very useful growth medium for identification of bacteria. It contains mammalian blood, usually 5% sheep's blood. It's considered a non-selective media because lots of different bacteria will grow on it. It's an enriched media because it contains blood for enrichment. And it's differential because different bacteria have different hemolysis patterns. Let's take a look at this slide down here in the corner. In the middle of the slide, someone has written the letter beta with a strain of bacteria, and you can see that there is clearing, white clearing around the letter B, and that's because of hemolysis. The bacteria plated here are hemolyzing the red blood cells and creating a zone of clearance around them, and that's what beta hemolysis looks like. Down here on the right side, there is what's called gamma hemolysis. These bacteria are growing on the media and there's no zone of clearance around them at all. So that's called gamma hemolysis, which really means no hemolysis at all. And then alpha hemolysis is partial hemolysis. It's somewhere in the middle between the beta and gamma patterns. And I've summarized this over here. Beta pattern means complete lysis of red cells. Gamma means no lysis, and alpha is somewhere in the middle. The catalase test is used to differentiate staph from strep, and the catalase enzyme is produced by bacteria that use oxygen, so it's produced by aerobic bacteria. And this is an easy way to remember that staph is aerobic because it has catalase present. Catalase breaks down hydrogen peroxide into water and oxygen, and the little oxygen molecules that are created will make bubbles. That's how you interpret the test. So what you do is you put some hydrogen peroxide on a slide and you add some bacteria and you look for bubbles. And there's an example of that over here. On the left side, we've got a catalase negative bacteria, so there are no bubbles. On the right side is a catalase positive bacteria, so we do see bubbles. There is a very rare condition tied into the catalase enzyme called chronic granulomatous disease. And this often comes up on the board, so I mention it now to help you keep it fresh in your mind. So phagocytes in the human body use an enzyme called NADPH oxidase, and this generates hydrogen peroxide from oxygen. This is a process called the respiratory burst, which you learn about in biochemistry. In chronic granulomatous disease, patients have a loss of function of NADPH oxidase, and this means that the phagocytes can't generate hydrogen peroxide in order to kill bacteria. Now, if a person has one of these loss of function NADPH oxidase mutations and they get an infection with a catalase negative bacteria, they will be okay. That's because the catalase negative bacteria generate their own hydrogen peroxide, which the phagocytes can then use despite the enzyme deficiency. However, if a catalase positive bacteria is present in a patient who has chronic granulomatous disease, that catalase positive bacteria will break down the hydrogen peroxide. This means the host phagocytes will have no hydrogen peroxide to use, and the result will be recurrent infections. And there are 5 organisms that cause almost all of the illness in patients with chronic granulomatous disease, and that includes Staph aureus, Pseudomonas, Serratia, Nocardia, and Aspergillus. And these are all catalase positive organisms. And you should know that Staph aureus is catalase positive because that's how we identify it in the gram-positive algorithm. You may not know that all the rest of these are catalase positive, but you should at least remember that Pseudomonas is, because that causes a lot of infections in patients with CGD. So the next test we'll talk about is called the coagulase test, and this differentiates Staph aureus from other staph, so-called coagulase negative staph. What you do is you put a bit of rabbit plasma in the tube and you add bacteria. If coagulase is present on the cell surface, it causes fibrin clots to form. So coagulase positive organisms like Staph aureus will show clumping of blood as clots form. Coagulase negative organisms will show no clumping. Next test is the novobiocin sensitivity test, and this differentiates Staph saprophyticus from Staph epidermidis. And the way you do any antibiotic sensitivity test for bacteria is you plate the bacteria on agar with a disc of the antibiotic present. In this case, it's a disc of novobiocin, and then you look for clearance around the disc. So let's look at this picture down here on the right. The white circles are discs impregnated with antibiotics. On the left side, bacteria are growing right up to the edge of the disc, and that's because they are resistant to that antibiotic. They're able to grow even in its presence. On the left side, the bacteria have a zone of clearance around the antibiotic disc. That's because they're sensitive to it and they can't grow up to the area right next to the disc. So bacteria that are resistant to novobiocin will grow near the edge of the disc, and bacteria that are sensitive will have a large zone of clearance around the disc. Next test is the bacitracin sensitivity test, and this differentiates group A strep from group B strep. Bacitracin is an antibiotic that interferes with peptidoglycan synthesis, and you may know that you can buy it as an ointment for the treatment of skin infections. Bacteria vary in their susceptibility, and the technique for using this test is the same as what we talked about before. You plate bacteria on agar with a bacitracin disc, and you look for clearance around the disc. So resistant bacteria will grow near the edge of the disc, and sensitive bacteria will have a large zone of clearance around the disc, telling you that the bacteria is sensitive. The next antibiotic sensitivity test is the Optochin sensitivity test. Optochin is also called ethyl hydrocuprine, and its use is to differentiate strep pneumo from strep viridans. So strep pneumo is highly sensitive to Optochin, and the way I've always remembered this is to imagine a little 2 hanging off the O in Optogen for O2, and that stands for oxygen, which reminds me of the lungs, and that reminds me that strep pneumo causes pneumonia, and that's how I remember that strep pneumo is Optochin sensitive. That's just always worked for me. Use it if you wish. The technique is the same as the technique for all the other antibiotic sensitivity tests. You plate bacteria on agar with a disc, and you look for clearance around the disc. If it's resistant, like strep viridans, growth will be near the edge of the disc. If it's sensitive, like strep pneumo, there will be a large zone of clearance around the disc. Next, we're going to talk about this group of bacteria down here, the so-called Group D streptococci. And they're called gamma hemolytic because they often don't hemolyze blood agar, but in reality, their hemolysis patterns are variable. If you leave them on the blood agar long enough, you may see some beta hemolysis or alpha hemolysis. And really, the way we identify these bacteria is often not by the hemolysis pattern, but by some other techniques we'll talk about now. So bile esculin agar is a selective growth media for both of the medically important group D strep. Both Enterococci and Strep bovis will grow on this medium. It contains bile salts which inhibit most gram-positive bacteria except for these two. It also contains esculin, which is a substance that's hydrolyzed by group D strep, and this turns the growth medium dark brown or black. So if you have a bacteria that is a gram-positive cocci and you put it in a tube of bile esculin agar and the bacteria grow and turn the media dark brown or black, then you know that you are dealing with group D strep. And this is the way it can be done in a micro laboratory. Once you know you're dealing with a group D strep from the bile esculin test, you can plate it on sodium chloride media, and this will differentiate Enterococcus from non-Enterococcus group D bacteria like Strep bovis. So Enterococcus is what we call salt tolerant, and it can grow in high salt concentrations. I just think of Enterococcus as a very tough bacteria. It's resistant to a lot of antibiotics, and it also can grow in high salt concentrations. So when you inoculate your bacteria in a high sodium chloride media, you then watch for growth. And if it grows, you know you've got Enterococcus. And if it doesn't grow, you know you're dealing with Strep bovis. So here's a slide to summarize the identification of the group D strep. If you get a positive result on the bile esculin agar, meaning it turns black, then you know you're dealing with the group D strep. If it then grows on sodium chloride, then you know you've got Enterococcus. And if it does not grow on sodium chloride, then you're dealing with a Strep bovis infection. The last bacterial identification test I'll mention before moving on to gram-negatives is that of mannitol salt agar, and this is used to differentiate staph species, so it's used to separate staph into Staph aureus and other staph. So this medium contains a high concentration of salt, and staph can tolerate these high saline levels. It also contains mannitol and a pH indicator called phenol red. Now mannitol fermenters like Staph aureus make acid, and the acid changes the phenol red to yellow. So Staph aureus ferments the mannitol, but other staph do not. So the bottom line is that growth on mannitol salt agar with a yellow color is Staph aureus. I've got a picture for that up here in the corner. So in the bottom there's Staph aureus growing, and the phenol red has been turned to yellow by Staph aureus. We've got some other kinds of staph in the other parts of the growth plate, and they are not turning it yellow, they are staying red. So I mention this because it is sometimes used to differentiate staph from each other. And also I mention it because I don't want you to confuse this with the salt test for group D strep. This is a very different salt test used for staph. It has nothing to do with identification of Enterococcus and salt tolerance for those bacteria. This is the algorithm for identification of gram-negative bacteria. So once you find out you're dealing with a gram-negative bacteria, you can look at the shape. So Diplococci are generally going to be Neisseria. There's one other bacteria they could be called Moraxella, but it's usually not speciated. Once you know you're dealing with Neisseria, you can differentiate those by whether they ferment maltose or they do not ferment maltose. There are a number of gram-negative Coccobacillus, which I've listed here on this slide. Some gram-negative bugs are what's called comma-shaped. These are generally going to be oxidase positive. We'll talk about that test. And then the two comma-shaped gram-negatives are Vibrio and Campylobacter. And Vibrio grows in alkaline media and Campylobacter grows in warm temperatures like 42 degrees Celsius. One of the most common shapes for gram-negative bugs is gram-negative rods, and we separate those by whether they ferment lactose or not on MacConkey's agar. I'll talk about that in a minute. Among those that can ferment lactose, there are so-called fast fermenters like these ones here and slow like these ones here. We'll talk about what those mean. And then once you know that something doesn't ferment lactose, the next test that's usually done is the oxidase test. And if it's positive, it's Pseudomonas or H. pylori, and if it's negative, it's one of these bacteria down here. We'll start by talking about the maltose test. So Neisseria meningitidis can metabolize maltose, but Neisseria gonorrhea cannot. So you use a growth media with maltose present to differentiate one from the other. The ability to ferment lactose is generally done by growing bacteria on MacConkey's agar. This is a selective media for gram-negative bacteria. It contains bile salts that inhibit the growth of gram-positive bacteria. It's also a differential media for lactose fermenters because lactose fermenters produce acid, and this will turn the agar pink. Non-lactose fermenting bacteria will grow as colorless colonies. And this is a picture of MacConkey's agar, and there are some lactose fermenters on the left side of the dish, and they've turned the media pink. On the right side are some non-lactose fermenters, and you can see that they are growing as colorless colonies. Some bacteria are what we call slow lactose fermenters. So Citrobacter and Serratia will initially appear to be non-lactose fermenting due to slow growth. But if you leave them in the incubator for a longer period of time, they will eventually show growth and turn the media pink. So these are called slow lactose fermenters, and that's one way to differentiate them from other faster lactose fermenters. The oxidase test is a test for the presence of an enzyme called cytochrome oxidase. So it's very simple to do. You take bacterial colonies and you place them on paper discs which have an indicator present for the presence of oxidase. And if it's present, you will see a color change occur. And then you know you're dealing with an oxidase positive bacteria. So back to our gram-negative algorithm, there are two groups of oxidase-positive bacteria you should know about. So the lactose non-fermenting rods that are oxidase-positive are Pseudomonas and H. pylori. But we also have this group up here that's oxidase-positive and comma-shaped, and we're going to talk about those right now. So Campylobacter and Vibrio are oxidase-positive organisms that are comma-shaped. And the way you can tell them apart is by where they grow. So Campylobacter will grow at 42 degrees Celsius, but Vibrio will not. And Vibrio will grow on an alkaline media, but Campylobacter will not. And then back to our algorithm one more time. The final test I'm going to mention is something used for oxidase-negative gram-negative rods. So you can differentiate this group further by using hydrogen sulfide testing. It's not shown in this slide, but it can be done to separate Shigella from Salmonella and Proteus. So the key point to remember here is that Salmonella and Proteus produce hydrogen sulfide, whereas Shigella does not. And you can use a test called the triple sugar iron test to look for production of hydrogen sulfide. Organisms that produce hydrogen sulfide, like Salmonella, will turn the TSI media black. Organisms that do not produce hydrogen sulfide, like Shigella, will not turn the medium black. So this is a simple test to see if the organism can produce hydrogen sulfide, and it's largely used to separate Salmonella from Shigella, because as you know, they can both cause enteric infections. And that concludes our module on bacterial identification.